So this is video 8 on coding with R. We'll look at basic plotting in R and some concepts behind the way we approach it. We'll also talk about the labs at the end of this video as well. So far we talked about the logistics of coding with R, talked about why we want to do it, the way we manage data with R in brief, how to run R commands, how to install the sort package so you can use it, variable vectors and operators, and data frames as well. R is really fantastic for plotting and it's great for data visualization. Over the years, many different approaches to data visualization have been used, and they're very, very useful for understanding diseases and outbreaks and weird infective agents. This is an example of I guess, a scientific figure from James Ferran and colleagues in 1885 made with oil paint. So that's the predecessor of advanced plotting in R or other languages in Valencia in Spain. And what this is showing is details on things like maybe the temperature over different months, April, May, June, July, August, September, of this nasty cholera outbreak. And this graph shows the rate of cases and deaths over time and other things like the temperature of the soil. So putting together this information and plotting it together, they can better understand what was going on, what was causing the outbreak, and what were the factors that made it worse or not as bad. Another way we can think about plotting with R is that we can, integrate different data sets, especially ones that are maybe over a period of time. We know that young infants are very prone to infections, and we know that measles outbreaks before vaccination was possible were very, very common, especially in younger children who are much more susceptible to it. We know that in London that there was high rates of infections before there was vaccinations brought in 1968. So you can see that intervention here. So initially, the observed cases, deaths of cases were shown here on this graph, showing the y-axis here. And you can see this is for different intervals over these different years. So note that this is death data first before switching to case data here at this junction about 1940 or so. So you can see that the case data as well is the square root because it would normally be much higher than this. But what you can see here, if you look at this graph, is that death rates, due to hygiene practices perhaps, were gradually going down, but there was periodic outbreaks all the time. And that kind of stabilized during the 1940s, 50s, 60s until vaccination came in to really reduce that to very, very low levels. So producing graphs like this are very, very possible with R coding and just takes a little bit of work to understand how to do it. One particular thing we want to avoid are these awful dynamite plots that you see all the time in biological graphs. These are horrendous, and we want to get away from this because they summarize data that you can see, you should be able to see as raw data. See that the bars here may represent a mean or median. The error bars here, that might be the standard error, the standard deviation, but typically on these plots, it'll never be clarified. And you don't know for these particular plots of these white and black bars, what was the underlying distribution. So you can see they could be like this, the symmetric options. They could be like this, where some of them have big outliers. They could be multimodal. They could be in different sample sizes, but this awful plot obscures all the information. And we want to move away from that as much as possible to avoid these issues where we really can't understand the underlying data. And that's the purpose of plotting, is to understand what we're looking at. And dynamite plots, or bar plots as they're sometimes called, are the wrong way of doing that. So if you think about this, if we have bar plots like this going off the white changing to black changing to white here, we don't know if all the different people in these conditions responded in the same way, or if they're very, very different, or which trajectories they take. It doesn't tell us. We don't know if the data is symmetric, if they're skewed, if there's big outliers, if they're bimodal. None of that is clear. And I just take this from a recent publication in the Lancet on the Oxford COVID vaccine group, where they're asking the question about higher IgG in older people. And they got the receptor binding domain, IgG titer here on a log scale, and they're grouping people. So they got a lump of younger-ish people, then people in their late 50s and 60s, and people over 70 lumped together, and they've got these fantastic p-values just labeled on top of the plots without stating what exact hypothesis is being tested, without stating what model is being used, and a complete absence of other details. So they're trying to make this claim here about the rates of decline in IgG levels in older people, but that's not what they're not visualizing this in the right way that can actually give you any confidence in what they're stating. But this is summary data. They might as well just make a table of the summary values here. These plots have no value. They don't tell us anything more than a table would do. You know, there's heterogeneity here across these different distributions that is simply not shown at all. So you can ask yourselves, why are they binning a continuous variable? Age is a continuous variable. You don't need to bin it into categories. It's the wrong type of approach to use. And this was in the Lancet. The model being used was not mentioned. What test is being performed, not mentioned. The standard errors are shown, but not the standard deviation. So we've no feeling for the underlying variation within the population, and that might be the more important result about the structure within the population. What numbers of samples? Clearly, they're going to have far more in this first category than in this category here. And that can affect the results or in the interpretation results and indeed the model that's being used. The y-axis scaled and it's cut here. It's cut down at the end, which is not good practice. And the sampling per group is highly likely to be imbalanced, and that really needs to be addressed. So we want to avoid these awful bar plots and think that there are many, many ways of creating nice visualizations that will make things much, much better. So in the labs, we'll talk about that in more detail. We'll address more clearly these aims of learning basic or data structures, data types, and inbuilt functions, and how to let or do the hard work for you. We'll read about data input and output. We'll learn more about sophisticated data manipulation to make things more efficient for understanding R. So that concludes this part on basic plotting with R. And please note in the labs, we'll be revising the key concepts of R that we're talking about here. We're looking at the practical aspects of vectors, variables, and operators in R. We'll look at data frames and tabular data. We'll look at importing data into R and exporting it as well. We'll look at base plotting with R. We'll look at how to avoid alternatives to these awful dynamite plots I just mentioned. We'll look at how to write blocks of code and use if and else statements to write conditional code so that you can deal with the data that you have. We'll look at for loops so you can repeat code many, many times and take advantage of these computer of the R environment. And then we'll also look more broadly at tidy data with R and how you should organize your data when you're doing a project involving large data sets or you're using code to analyze it. So that's the end of this video.